So we'll start recording. I was looking at the syllabus and noted that this is week 13. So we hope to, according to the schedule, wrap up chapter 13 and begin chapter 14. With our third exam scheduled for next Tuesday, or Thursday rather, the 29th. So I wanted to talk just a little bit about that, even though we're a week away. It doesn't hurt to be proactive and be thinking about what's coming up in the next few weeks. So if you recall, exam 2, which we had back on April 1st, covered chapters 6, 7, and 8. So exam 3 will basically cover chapters 10, 11, and 12. And then we'll put 13, 14, and 15 on the fourth exam. Okay. So exam 3 next week over chapters 10, 11, and 12. We also have a quiz scheduled for this week on the 22nd. And I'd like to have that be over chapter 12, which I know is a big chapter, but you should have gotten through that by now. Actually, let's maybe focus in a little bit. That is such a huge chapter for just one quiz. Let's do this. Let's have the quiz this week cover sections 12.4 through 12.6. So 12.4, 12.5, 12.6. I think it's unfair to give you a quiz over an entire chapter. Gosh, it's just such a huge chapter. Everybody hear that?
Yep.
Okay, great. Thank you. All right. So I think we've got all of our housekeeping items done that I could think of. Am I forgetting anything in terms of quiz, exam next week? One thing that I wanted to mention, we're, as I said a moment ago, supposed to be wrapping up chapter 13 and starting 14. I have one more lecture, Zoom lecture, to make in chapter 13 on the epidemiology section. I think there's like maybe 2 sections on that. And my goal is to do that tomorrow afternoon, just in case you're wondering about chapter 13 and that. So if I asked you, how many of you are into chapter 13? Maybe half the hands go up, maybe, if I'm lucky. I don't know. Anyway, we should be done with that chapter in terms of Zoom lectures. I'm hoping to get that done tomorrow afternoon. Now, you might notice that I've already put up the Zoom lectures for chapters 14 and 15, which are the last 2 chapters of the book. And what I did for that is I took what I had recorded last spring and I edited and snipped and pasted that together. So that actually came from last spring. And so you're going to see some students in the upper right-hand corner of the Zoom lecture from that semester. So I wanted to give you a heads up on that. So I'll open the floor. Questions about what I just have been talking about, or we can talk about, chapter 10, 11, 12, 13. Not sure where you are. Hopefully you're into chapter 12 by now.
Could you go over retrovirus therapy? Is that what it's called? Yeah, in 12.
Okay, sure. So let's see what section that was. Gabby, do you have a particular section here or page?
I'm looking at the PowerPoint and it's slide 55. I'm not sure about the book.
Okay, thank you. 55 is great. So let me pull that up. Was that this one? Oh, I gotta share, don't I? Here, sorry. Okay. All right, let's see. This is slide 55.
Yeah, that one.
Okay. This was not so much a slide that specifically talks about the role of retroviruses as therapeutic agents, but just sort of reviewed how retroviruses worked. And because historically most people know about HIV and AIDS, I often link those two things, the actual virus and the disease, with retroviruses in general and how they function. So is your question, how are retroviruses used therapeutically, or is your question, how do retroviruses work?
I guess like how they're treated. Like I think on slide 57, do we basically just need to know that like the reverse transcriptase is inhibited?
Yeah. Okay. So
Okay.
so what I was talking about in these slides you're quoting is how do certain types of drugs like interferons and AZT specifically is the one that I think of, but there's been a whole slug of them that have been developed in the last 20, 30 years. But when AZT was one of the first anti-AIDS drugs prescribed. And since that time, they've come up, as I said, with a whole slug of them, and they're used in what is sometimes referred to as a cocktail of drugs that are given to an AIDS patient. And I think I may have mentioned in some of the earlier Zoom lectures that, well, HIV back in the mid 80s, when it first kind of came out publicly, was thought to be pretty much a death sentence. If you got AIDS in about 10 years later, post-infection, you'd probably die. Turning that into now a much more chronic, well-managed disease where there's very little, I shouldn't say very little risk, but your odds of living a long life with AIDS are very likely today. So I think while we haven't cured it, we've come a heck of a long way in 30 years. So what I've got focused here are these two sort of blue ovaled 5 and 6, right? These are drugs that act to inhibit the ability of the retrovirus, HIV virus specifically, to prevent the replication of the DNA. So you remember retroviruses use this reverse transcriptase that they bring with them into the host cell, and then that is used to take and synthesize DNA from the messenger RNA. And it's called reverse transcriptase because it is the reverse of transcription, right? Which is DNA to messenger RNA, the first step in protein synthesis, right? So this is a little trick that retroviruses have up their sleeve. They can take and have the cell synthesize viral DNA and then replicate that for the virus. And then that DNA can be used in the normal transcription translation process to make more HIV protein and also make more HIV RNA. So what these drugs do is they get in there and they inhibit the action of the enzyme. That's what this number 5 is talking about, the nucleoside analog reverse inhibitors. The names of these drugs, hard enough to pronounce, much less remember, right? I mean, they're crazy names. These are designed by people in pharmaceutical companies, and I don't know how they come up with these words, but they do. So anyway, here we're looking at the inhibitor as a pink, kind of a little, I don't know what you call that, little house-shaped symbol, and gets into the helper T cell, which is really the cell that's targeted by the AIDS virus. You might remember that from ANP1. It's helper T cells. Oh, here it is, right here. Comes into the cell, and again, it prevents the reverse transcriptase from working, from functioning. Again, the manufacture of DNA from messenger RNA. And the non-nucleoside reverse inhibitors. So this is called nucleoside, this is non-nucleoside. It doesn't necessarily impact let's see, doesn't so much block the viral DNA synthesis from the messenger RNA, but rather, as it says here, attaches to the reverse transcriptase binding site, not allowing it to work. I guess it's the same thing as blocking the action in that sense. But either way, you shut down reverse transcriptase. That's all you really need to know. We talk about some of these protease inhibitors here. This has nothing to do so much with reverse transcriptase, but rather, as it says here, inserts onto the protease enzyme, the AIDS virus protease enzyme, that would ordinarily be used to edit the protein that becomes part of the viral particle itself. So if the protease can't tear apart, clip the various proteins and have that then assembled into, say, an envelope of the virus, then you have a non-functional virus. It may actually leave the host, like we're seeing here, the host cell, but because it's lacking important protein components, if you will, within the nucleocapsid, it can't go on and infect other helper T cells. So the point is that you can attack, if you will, the HIV virus in a number of different ways. You can affect its ability to synthesize DNA from messenger RNA, i.e. target the reverse transcriptase enzyme, or you can, in this case of the protease inhibitors, prevent the normal construction of the outer protein coat of the virus. Either way, you're manipulating the ability of the virus to do what it needs to do inside the host cell. And that's how a lot of antivirals work. They capitalize on important critical biochemical steps, if you will, or processes that occur within the host cell. And if you can affect those and screw them up, the virus can't complete its life cycle within the host. Or even if it's released, it can't go on to invade other cells. Does that help provide some clarity, or did I muddy the waters even worse?
Yeah, that was good. Thank you.
How about other questions on chapter 12?
I have a question.
Yeah.
So if you go to slide 13, there's a chart and it talks about the different effects of like, you can see it. Do we have to memorize like all of those different medications and their effects?
Okay. I know this looks like a really daunting list. I will say that certainly for the Helminth drugs, now these are the parasitic worms, right? You do not need to know the names of these. I think for fungal infections, I would know amphotericin B. I'm not concerned about these other two. For viruses, we just talked about AZT. So I think you ought to be familiar with that. Azithromycin is what it's called, AZT. I don't think you need to know the names of all the other drugs that are listed in that table, I think they call it, or a figure. However, when we get to the bacteria here, I pretty much go through and I talk about, I think, every one of those, with maybe the exception of, well, no, I think we go into almost all of those. Each one of these has its own specific set of slides. So I think you ought to be familiar with those. I know it looks like a lot. Just remember that these various antibiotics, most of these are antibiotics, are targeting one of 5 different cellular processes or structures, right? And of course, we can see that under the effects of the drug. But if you go back to this one set of, here it is, this diagram. This may help a little bit. What you could do is reference this in your mind's eye, maybe, to help you kind of remember which drug impacts what aspect of the cell, whether it's how the cell wall is constructed, assembled, right? Or how the cell membrane functions, or how the cell makes protein by targeting specific parts of the ribosome, or talking about how metabolic pathways are impacted. A lot of the sulfa drugs do that, right? I mean, I really think that this figure, while they don't use it in this particular edition of the book, I think I told you guys last week, for some reason they did away with it and they went with this. Right? Remember me talking about that? This is
Yeah.
good. I don't mind it, but I like the old one. Call me old-fashioned. I don't know. It's not that I was unwilling to put a new figure in if I thought it was better than what I had, but to me, I think this is better. So, what you might want to do is go back to that table. Where was it again? Slide what?
13.
13, thanks. Compare this slide with this diagram and cross out any on here that don't appear over here. There's going to be a lot of the same thing, obviously, but as I look at this diagram here, like this, you don't have to know this or that one or these 3. These others, I think, appear on that table, don't they? Pretty sure they do. This is where having, a poster board maybe, or a sheet that's devoted to cell wall inhibitors, another sheet devoted to protein synthesis inhibitors and so forth might be helpful. I do understand your concern. I mean, I know that's a lot. But a lot of you guys are going into nursing, you're going into health-related fields. Do you need to know that tetracyclines inhibit protein synthesis? They're not going to ask you that only in general. I know, I get that. But you're going to run across some of these drugs. Some of you already have in your work. And now you'll have some sense of what they do. How do they inhibit bacteria?
This is what I'm in the 4th semester of nursing, and this is what we're learning about right now, like drug therapy for infections, viral, fungal, and parasitic ones. So
Oh,
a lot
wow.
of it lines up.
Oh, that's good to hear. You know, and I haven't talked with the nursing folks, and I should probably, just because we do want to kind of support one another in terms of what you guys are hearing. So I'm really, really happy to hear that. So are many of these the same sorts of things you're talking about in nursing?
Well, it's the pharmacology class. Well, you
Oh.
have to take it with it anyway.
Yeah.
So yeah, it's lining up a lot. Just because I was reading it today, and I was like, oh, like there's a whole section about like the helmets thing, and I was like, oh, this is the first I've heard of it in nursing, but okay.
Yeah. Pharmacology, that's 3 credit hours, isn't it?
I think so. Yeah.
Mm-hmm. And would you say that having micro before hearing about it in nursing is preferable, or how many of you, well, I don't know how many of you are in the same boat, but are some of you finding the same situation whereby what we're talking about here in chapter 12, you're also learning about in the pharmacology course? No?
I might be the only one that's in the 4th semester. I don't know.
Yeah. So are you graduating next?
Well, this, yeah, in 26 days, yes.
Oh yeah, great. All right. So is it safe to say then it's likely that most people in pharmacology have already had this in micro, do you think? Or you're not sure?
Probably. I mean, I'm taking mine kind of late because I took all of my prerequisites with nursing.
Yeah, right.
So
Yeah.
I'm guessing, yeah.
Yeah. Well, I guess lucky or unlucky for you, you're getting a double whammy at the same time.
Yeah, it's a great time.
Yeah, I heard the nurses have a big pediatrics test coming up in 2 weeks.
Yeah, that was I think that was last semester and it sucked.
Yeah.
But just study.
Yeah, I study hard. That's what I hear. Okay. So yeah, that's the downside of this chapter, is kind of trying to remember what drugs do what to the cell. I fully get that angst or anxiety that can create. But I think a real big chunk of this chapter is really kind of understanding what are the different types of penicillins or antibiotics. We should just broaden it a bit. And exactly how does each group target specific aspects of the microorganism. But a big chunk of this right-hand part of table 12.4, you do not need to know the names of the drugs. But other questions? Are you finding chapter 12 to be a real slug to get through, or tough, or slog, I should say, to get through, or is it making sense? I know there's quite a bit of chemistry that I do make mention of. Don't worry about having to draw this or, really get into the chemistry here so much, and just understand kind of the fact that these cephalosporins do have they call it a common nucleus. It's kind of a funny name, nucleus, but it's kind of the common core of the molecule, and that we can then slap on different R groups to either side of this nucleus to create a whole host of these cillins, or not cillins, but KEF antibiotics. The penicillins are the illins, and likewise, they have a common nucleus too, and we just slap on a single R group on one side, and then we can create a whole host of additional cillins, close cousin to penicillin. Other questions, comments? How's this chapter going? I guess is what I would ask. Is it okay? Is it just a
I like
lot? Pardon?
I like it.
Do you? Yeah, I
Yeah.
think.
I
Why
think
do you
it's-
like it, Monica?
I don't know. I think 12 and 13 are probably my favorite so far, as far as interest.
Yeah.
But
good.
I was wondering if maybe if no one has questions, if we could go over maybe what's going to be on the quiz, like those sections, just to hear it again.
Oh, yeah. So let's go back. This was chapter 12, which we're in, and that was starting with section 12.4, which was interactions between drugs and microbes. So that gets, I think, to this. Do you all see the acquisition of drug resistance slide? Or no?
Yeah.
Okay.
Yeah.
Okay. So what this is talking a little bit about is this idea that when you alter the genes within the DNA, sometimes that can be beneficial to a population of bacteria. And so this number one, acquisition, spontaneous mutations, is getting at sort of processes that occur in the genomes of all organisms, not just bacteria, but every single living cell population. This is going to happen. And remember, in one of the earlier chapters, we sort of said, while most of the time we think of mutations as being bad, right? It doesn't have to be bad for a population of organisms, because once in a while, that mutation may prove helpful to the population. So you've got to think of bacteria now, even though they're individual cells, you have to think of them as just one cell among many trillions of cells in a population. A population is defined as a single species of organisms of the same type. Okay, so maybe all E. coli, all Klebsiella, all Bacillus megaterium, whatever species you want to think of. In that population, there'll be some cells that, as the DNA replicates during cell division, you're going to get occasional alterations, random, spontaneous mutations. And once in a while, once in a blue moon, that can be very helpful, and that can actually promote drug resistance characteristics. If that population is subjected to a particular drug, and it has now the ability to do something it couldn't do before, right? Code for a different protein, modify a given protein a little bit so that it's more anti-drug characteristic is improved, then that can be helpful to the cell, because the cell survives the drug, it reproduces, those cells that come from that cell all have that same mutation. So this, as I mentioned, is the most common cause of drug resistance. It's just normal evolutionary processes that I cannot control. Medicine cannot control that. It's going to always happen. I mean, what are we seeing today, for example, with COVID-19? I know we're switching gears here a little bit, but let's just chat for a second about that. What have you been hearing the last few months about COVID-19? What are some concerns?
There's a bunch of different variants starting to pop up.
Yeah, exactly. Well, what does that mean? It means that the original COVID-19 virus, whatever we want to call it, the original, has undergone this very process. That's what we're talking about. Yeah. It's the same idea. Okay, and then the other thing noted here, of course, is this ability of bacteria to get new genes from other sources. And we talked about this in, was it chapter 11 or 10? I just cannot remember. Maybe it was 9. Yeah, it was 9, chapter 9. At the end of chapter 9, we talked about transformation, transduction, and conjugation. Do you remember that? Yeah, yeah. So this second source of acquired genes is nothing new. You should look at that slide and it should make some sense to you. We're not introducing anything new here. Are we good with that? Oops, sorry. Any questions on that slide? Okay. So what the next few slides talk about are ways in which drugs specifically are circumvented, if you will, by different tricks that bacteria have at their disposal to negate the action of the drug, in other words. How is the drug resisted, if you will, by the cell? Well, let's look at the first one here. Here's penicillin. And some bacteria have an enzyme called penicillinase. Look what this enzyme does to the penicillin. Well, you can see it breaks this bond here. We create an inactive form of the penicillin. Okay, so there you go. There's negating the action of this very, very, very important antibiotic. And what can the cell not do then? Or what can the cell do, I guess, is what I should say. What can the cell do now that there's no penicillin to affect it? It can make what? What does penicillin target?
Is it the proteins?
No. It affects the cell wall and how it's constructed. Page 375. So now the cell can make more peptidoglycan as it undergoes binary fission. If it didn't have the penicillinase there, the penicillin would prevent that peptidoglycan formation and the cell couldn't divide. The second technique here involves altering the receptor site on the surface of the bacterial cell. So under normal circumstances, we've got a drug shown here in blue that docks with special receptor sites and is allowed inside the cell where it harms the cell. Well, what does the cell say to that? It says, okay, fine, I've got the drug here outside me, but guess what? I'm not allowing it in. Why not? How do I prevent that? I change my surface receptor. It can't combine, it can't be allowed in. Now, these surface receptors are really what kind of molecules? Are they carbohydrates, lipids, proteins, nucleic acids?
Protein?
Right, they're protein receptors. Yeah. So how does a cell make a protein? Well, it undergoes transcription and translation, right? Both of which start back at the DNA level, right? The transcription of the messenger RNA from the DNA. Well, do you see what's going on here now? If we alter the DNA, what do we then also alter? The messenger RNA. What then happens? We make a different what?
Protein?
Right, and there it is. There's the different protein. So do you see can you connect the dots here? Altering the DNA, changing the genetics, either through acquisition of genes from another cell or maybe through spontaneous mutations, allows me to make a different protein here on the surface. And now that is beneficial to me as a bacterial cell because the drug can't hurt me. This is another really interesting one. The drug is allowed in, but we activate certain special protein pumps that pump the drug right back out. Isn't that crazy? If we get rid of the drug intracellularly, it can't affect the ribosome. It can affect various metabolic pathways, whatever the drug's action is. If it can't be there to do its work because it's getting pumped out, hey, I'm happy as a bacterium if I can do that, right? Oops, sorry. Number 4, change in drug binding site. So here we're looking at a drug. And if we go to that table, well, actually to that sketch, I'm not going to have that in the book. Anyway, there's different kinds of antibiotics that specifically attach to the ribosome here, for example. So here's the normal functional antibiotic in green. It locks into the large ribosomal subunit. The small subunit can't connect to it because of its action. It prevents protein synthesis, right, normally. But what does the bacteria do? It says, I am going to alter my large ribosomal subunit a little bit so that there may be plenty of drug here, but can it bind here? Can it attach like it did over here? The answer is no. If it can't attach, it can't prevent these two subunits from linking together. So they link together and we continue our merry way and we translate the protein, the bacterial protein. So this drug is inactivated because the ribosome structure has been altered a bit. And then in the fifth one, here, number 5, we're saying ordinarily, let's say, the pathway of a particular series of metabolic reactions involves, A converted to B, converted to C, converted to D, and blah, 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 until we get some end product. And the drug prevents conversion of B to C. Sulfa drugs, for example, often do this. Well, what does the bacteria do? It says, okay, fine. I'm going to go around you. I'm going to create an alternate pathway. I'm going to make C1 from B, and I'll make D1 from C1. And guess what? I'm going to make what product I need. That's all I care about. I'm going to go around the action of the drug, kind of like a bypass. So these are just 5 examples of how drugs can, how bacteria can negate the action of drugs, how drug resistance can develop in populations. There are, I'm sure, many, many, many other mechanisms. We just wanted to introduce a few of them so you have a flavor for how this works. It's pretty cool. And then I do have a video that goes through these. And I hope you had a chance to watch that. If you haven't, you definitely need to make sure you watch that. I'm not sure why this is a red M. This gets at the idea of natural selection and evolution, meaning that in a given population of bacteria, you're going to have some cells that are naturally able to resist the action of a given drug due to, again, a mutation or the acquisition of genes from other cells. It's only when we expose this population to a given drug that we see how important these two guys are, right? Because obviously they're surviving the action of that drug. What happens to all the other ones here, or the vast majority of them? They die, right? Well, these survive, they reproduce, and in a short period of time, suddenly we now have a very altered population. Those that were infrequent, if you will, or in a low population, statistically, numbers-wise, now can become the majority. The minority become the majority. Why? Because we've exposed that population to the drug. And if we continue to do that again and again and again and again for years and years and years and years and years, this doesn't happen overnight, this whole process, but it can happen fairly quickly. We got to make sure we don't overprescribe drugs, because if you overprescribe drugs, this process is accelerated, right? That's why it's often not a bad idea in certain instances to take drugs and shelf them, put them back on the shelf, if you will. Take them out of circulation, take them out of what's the word? Don't dispense them with near the frequency that you used to do. Because if you continue to use the same drug again and again and again and again, you're going to get to a point where it's no longer going to work, or the toxic effect of the high concentration or high dose is worse than the symptoms of the disease. That's what happened to penicillin back in the 40s. Penicillin was used widely during World War II to treat all these wounds that the GIs had. And it was wonderful. It was great. I mean, my gosh, it was like a miracle drug. But if they kept using it so much following World War II that it got to a point where the toxicity levels, they had to use such high dosages as time went on because of this, that it was causing more harm to the tissue than it was the benefit of the infection. So they had to pull it off the shelf and start using it less and less. So that's sort of the theory behind that. And that's kind of what I just talked about. Again, I think this table kind of makes sense if you just kind of read through strategies that pharmaceutical companies sort of have to be mindful of as they make these drugs. The importance of following directions the physician gives you for taking a drug. If you're given a drug to take for 5 days, 1 pill a day, don't stop after the second day because your sore throat's gone and think you can just put the 3 pills in the bottle and then pull it out in a year when you've got a sore throat again. People do that. If you do that, if you don't complete that whole dosage, you are really subconsciously promoting drug resistance. You want to be able to kill all those cells. If you don't kill them all, some survive. They're going to reproduce. You're going to continue to promote that drug resistance. So I think you should be able to read through this, and it's fairly self-explanatory. This thing down here at the bottom, this third bullet under long-term strategies, this is pretty cool. The addition of antimicrobials to animal feed should be curtailed, must be curtailed, not should be, must be, because what they're finding is that they're using so many drugs and antibiotics to treat cows and pigs and chickens and turkeys and everything that we love to eat, to try to keep them healthy so we can get them to market quicker, so we can make more money faster. It's all money. What we're doing is we are promoting drug resistance. And we're also turning out a product, i.e. meat, that is so laden with antibiotics, who knows what effect that might have on you or I. I mean, that's a whole nother, I don't even know how to begin to study that, but it could have major implications on our health. This just begins to talk about adverse reactions between certain drugs and target tissues. So I think that's, again, pretty easy to read over. Not a whole lot of people have allergic reactions, but a certain percentage of the population will always have reactions to any antimicrobial. I don't care if it is the Pfizer vaccine, or now we're of course seeing in the press in recent weeks, the Johnson & Johnson has been shelved, temporarily because of the increased blood clotting they're finding in women, right, between 18 and 38. And not to diminish that, because it's important that we have safe drugs, but it should not come as a surprise to anybody that you're going to get some individuals that have sometimes serious reactions to the drug. I mean, a couple of women have passed away, have died because of it, right? And I don't at all mean to diminish or demean that whole thing. It's important we are safe. But I think epidemiologically, if you think about how many people will have been, say, vaccinated using the Johnson & Johnson vaccine. I mean, we're talking tens of millions of people. And you've got a dozen, 2 dozen people that have had allergic reactions to that, sometimes severe. What do you think? Should we stop production? Should we be doing some research? Should we continue on in vaccinations? I'd like to just hear your thoughts on that. We have some time to kind of talk about that because that's an interesting discussion to have. What do you think? Have you thought about
Well,
this?
well, as of right now, we have pulled it, and we do have two. We have the Moderna vaccine and stuff like that, which have similar effects. I know with those ones, you have to take more than one. You have to go for more than one round to do it. But if we have safer alternatives, I see nothing wrong with pulling the vaccine if we have safer alternatives.
Mhm.
And they can put it back on the market once they work those out.
Yeah, a good point, Samuel. Yep. Anybody else? Thoughts, comments?
I think it's probably for the best that they pull it to figure out what common denominator those women have had that have caused the blood clots. Because like Sam said, we do have the other 2 vaccines that are effective. And once they figure out what that common denominator is and say, if you fall in this area, try to get one of the other ones, put it back on the shelf.
Mm-hmm.
I mean, I do know that they have come out that the benefits do outweigh the risks of the potential of having the blood clots. And I mean, that's something the individual person would have to talk with their doctor about, I think.
Mm-hmm. Yeah, good point. Good points. Any other thoughts, comments? I agree. If we have alternate vaccines, which we do here in the US, right? We have more than enough Pfizer and more than enough Moderna to make up for the Johnson & Johnson shortage. But I'm going to just play devil's advocate just for a moment, okay? And I'm not disagreeing with anything you said. I do agree with what you're saying. But what if you are Czechoslovakia, or what if you are Vietnam, or what if you are Germany or Sweden, and you've got one vaccine, and that's the Johnson & Johnson. You don't have access to the other 2 like we do. Maybe because we bought them all up. I don't know. But I know it's true that in many countries, especially in Europe, this is the primary vaccination, other than the AstraZeneca, I think, is also one of the other ones, but it's also running into similar issues with regard to some blood clots, right? And you're the health professional, you're the epidemiologist for Austria, let's say, or whatever, and you've got your incidence rate of infections going up, right? We have a surge going on in some parts of the world, right? If I'm that person, I'm like, yeah, I get the need to do our due diligence and be careful. I totally get that. But if it's 98.9 or 99.7% effective, I'm throwing those numbers out as just numbers. But if it means I can get shots in the arms of people to increase herd immunity, meaning, right, 80% or above are vaccinated, right, against it. That's what herd immunity is. Do you see the quandary that some countries are in, where they're a little bit anxious about this? If people get concerned about, is it safe? I'm not going to get a shot, because they hear about this, and it's not like they shouldn't hear about it, but it's an interesting moral, I don't know, moral health dilemma. Yeah. You should think about this. It's pertinent to what we're talking about in this course, right?
Well, I suppose at that point, you just have to ask yourself if the ends justify the means. That would come down to just the morals of, every individual world leader and what they believe on that. I had a point. I was going somewhere with this, but I've forgotten.
No, that's all right. It'll come to you.
I think it needs.
Go ahead.
I think it needs to be left up. I think they still should provide it for the citizens in their country and leave it up to the citizens if they're willing to take the potential risk of developing that side effect.
Yeah, I see nothing wrong with that.
Yeah, you can't mandate vaccinations. I just don't think that's the way to go. But it's certainly interesting to think about anyway. We've talked about superinfections already, so I don't think I really need to talk about that unless there are questions on what that means. Everybody know what superinfections are? You should know about superinfections. This simply gets to the idea that you treat the primary infection, and here they're using the example of strep throat. Okay, so there's the infection. That's the locus of infection. That's where it's set up in the pharynx. But let's go down to the gut here, into the intestine, and we'll see. I'll connect the two in just a minute. But look in the intestine of how that healthy individual, other than having a sore throat, he has no trouble gastrointestinally, if that's a word. And in his intestine, small intestine, he's got a whole slug of residents here, right, that are normally found in the gut. And those normal floral bacteria keep possible transients, possible pathogenic transients in check, preventing them from getting out of line, i.e. massive cell production and toxicity, or pathogenicity, I should say, would result. But now you treat the sore throat, okay? You treat the strep throat with an antibiotic. Does that antibiotic just go here when you swallow it? No, it gets circulated throughout the entire body, right? So yes, you'll knock out the sore throat perhaps, but guess what might also end up happening? Well, you're going to kill possibly some of the normal flora here. And what happens to the pathogenic transients? They're no longer kept in check, right? They reproduce, and then pretty soon now you've got diarrhea or some other GI issue. So that's what we talk about, what we mean when we say superinfection, that treating one can have sort of a ripple effect, if you will, by altering the normal balance. This is the same basis for yeast infections in women, right? Lactobacilli normally keep the yeast in check because of the acids that are produced, but you take an antibiotic for a sore throat or whatever, it kills a lot of the lactobacilli. The yeast do the same thing as the blue squares, right? Pretty soon you got a yeast infection. Very common, right?
Now, why does that happen with some people and others it doesn't?
Yeah, it might have to do with the individual uniqueness of the microflora of the vagina. That's just a guess. I don't know the medical reason for that, but that's a supposition. Interesting question. Not being a woman, I don't have a sense of how common it is other than from what I've heard.
Well, like my mom, she'll take an antibiotic, and every single time she does, she ends up with a yeast infection. Now, I can take that same antibiotic the same amount of time she does, and I will not have an issue.
Yeah. But have you ever I'm not to get personal here.
Yes, I have.
And as a result of taking an antibiotic of a different kind?
No.
No. Okay. Just out of the blue, not sure what caused
Yeah.
it. Okay.
Yep.
Yeah. So tell me, you X chromosome people, how many have experienced yeast infections as a result of this phenomenon of taking an antibiotic for a different infection? Anybody willing to share this, or maybe I'm being too personal? I don't know.
I have.
No, it
Yes?
happens to me every time. Every time I take an antibiotic.
No matter what antibiotic it is?
Correct.
Oh, that's interesting. Anybody else want to comment?
I know I have. I just can't remember what antibiotic it was.
Yeah. So it is really probably safe to say, and again, I've just heard this anecdotally, that it's not uncommon for yeast infections post antibiotic regimen causing it. But like Hillary or like Tiffany said, doesn't necessarily mean everybody's going to have it. But then here's Jocelyn says, no matter when I take it, I've got to contend with that. So it's interesting, isn't it? Everybody's unique. And I suspect it's a uniqueness in the natural microflora, probably to some extent. Carolina writes, is this similar to the C. diff infection since it is caused by the overuse of ABT? I don't know. What is ABT? It's obviously a drug of some sort, right? Even if it is an antibiotic, yeah. I do not know the answer to that question. It wouldn't surprise me if there's a correlation.
There is. A lot of times they actually prescribe Bacid now with an antibiotic to prevent C. diff because it just takes all your normal flora out of the intestine and they end up with C. diff after being on antibiotic.
Yeah, Right, I'm just reading the chat here. I haven't been keeping up on that. Yep, very good. Okay, so let's see, where were we here? Okay, this gets to some very specific side effects of certain antibiotics. You do not need to memorize this table. I think it's just sort of interesting to see how some of these may be fairly innocuous for the most part, like an allergy, like we saw in the photograph in one of the earlier slides, to some stomach upset or diarrhea. But some of these, heart arrhythmia, seizures, these are pretty significant. Hemolysis, this is rupturing of red cells, reduction in the number of platelets, which of course can have ramifications on hemostasis, the stoppage of bleeding, damage to kidneys. So, these come with trade-offs, right? Like anything, it's never a sure thing. You just have to be mindful of this and how often the patient is taking a given drug, how strong the dose is, what's the health of the patient, how old. I mean, there's a zillion variables that need to be considered. This is not a be-all, end-all. But these are apparently some common side effects of different kinds of drugs. Interesting. And then we get into some considerations for choosing what sort of antibiotic or antimicrobial drug we want to use. We're going to be getting into this topic actually next week in lab, so it's just like perfect timing. What this is showing and what I talk about later here in this chapter is the use of antibiotic impregnated discs that are added to a Petri plate to which a particular species of bacteria has been inoculated. Morning lab, right? We just did this. Afternoon lab in another hour or so. We're going to be doing this, and we're going to see the effectiveness on different kinds of antibiotics on that particular species of bacteria. So E. coli, whatever you have spread onto this plate, you can see of these, 2, 4, 6, 7 discs, looks like E is showing the most inhibition, followed by B and G. Looks like C and A haven't had any impact. Of course, the inhibition is indicating the death of the cells, right? So this is a really kind of interesting visual way of assessing how different antibiotics specifically interact with specific species of bacteria. And as we said a moment ago, assessing the condition of the patient is super critical. Are there any underlying health issues? Is it not to go back to our topic again too much, but it's interesting that these blood clots with the Johnson & Johnson vaccine are impacting all women, right? Of reproductive age, I think, if I remember the zone. Interesting, isn't it? Yeah.
I wonder if they're on some kind of like birth control that's causing a side effect.
Yeah, right. Because if you look at the birth control pamphlet, which nobody ever reads, it specifically addresses heart issues and problems. Yep. I remember seeing that. And so this kind of goes through, as it indicates, a little more specifics as far as how you assess susceptibility using these antibiotic impregnated discs. This is called the Kirby-Bauer method of analysis. This is sort of interesting here, this synergistic effect whereby the combined efforts of CTX and AMC antibiotics is showing, a stronger reaction in terms of inhibiting the growth than either one alone. It's called synergistic, or the synergistic effect. Synergy meaning combining forces, if you will. This is cool. Here we're looking at drug-resistant colonies on the very edges of these zones of inhibition. Isn't that interesting? So it kills a lot of the cells, but here's some that survived and now are beginning to reproduce. Remember, this colony is not just one cell. Gosh, I don't know how many cells we got here. Millions, maybe. Yeah. So that's not something I've ever seen. I've done the Kirby Bauer stuff for years, but I've never seen this antibiotic resistance like you see here. That's very, very interesting, I think. And so you literally take a ruler and you measure this zone of inhibition in millimeters. You measure this whole diameter all the way across, and then you can compare these lengths that you measure to prescribed tables where they will say, in order to be considered resistant, it has to be equal to or less than 17 millimeters, or I'm sorry, less than 17 millimeters. To be sensitive, it's got to be equal to or greater than 22 millimeters. The bacteria is said to be sensitive, but only if you have 22 millimeters or bigger zone. So you'd measure these numbers, you'd measure the distances here, and then you'd go to a table and they would tell you whether it's resistant or susceptible or even intermediate, kind of not really one or the other. It's kind of like, we're not sure if it's sensitive or resistant. And then these are some other additional techniques. I've never used these e-test diffusion strips. I think it would be very cool to do. So what this is rather than having, 7 or so 8 different discs that you put on a single plate, you put a bunch of strips. You can kind of see the outline of the Petri plate here at the top. And each one of these strips has a given antibiotic in it. So IP is one, I don't know what it stands for. TZ is the other antibiotic. And then these numbers represent the dosages. So very, very diluted dose here at the bottom and much, much stronger as you get toward the top. And you can kind of tell where the minimum inhibitory concentration is. What's the minimum amount to inhibit growth? So like 1.5 for IP and about the same, isn't it, for TZ? Interesting. So this gives you some quantitative data. You can come up with MICs. I think that'd be fun. We should probably get some of those and just try them. I think that'd be cool. This is using broths that have had different dosages of a given antibiotic added to them. And that's what these numbers indicate. These are microgram dosages, and it looks like we're getting ever increasing, right? 0.1, 0.2, almost doubling. Yeah, actually we are doubling each time, aren't we? Yeah. And then we're inoculating, and if we see turbidity, what does that tell us?
There's still growth.
Right, and where it's clear?
There's no growth.
Exactly. So what's the MIC for this? Minimum inhibitory concentration.
(1.6).
Right. See how you do that? This is a little more labor intensive. You got to do a bunch of broths, and you got to, dispense the dosage into each broth. It's just a little more time consuming. It works fine. You can manipulate the concentrations to your desire, whatever you want to do. So it does have its positive upsides. And then this is just another product that they sell. Again, this is a bunch of little wells, little bitty tiny wells that have had different concentrations of different antibiotics added to them. Okay, so this is low dose. As the numbers go up to 10 or 12 rather, it gets to be higher doses of the given antibiotic. And again, I'm not sure what all these abbreviations stand for, but they're antibiotics. And then somehow a machine probably inoculates a small quantity of the bacteria to each of these 12 times however many we got here. 2, 4, 6, 8. So 96 little wells get inoculated by a machine and then incubated, and then you pull it out. And the pink, as it indicates here, is where we got growth. Blue indicates no growth. And the X's here are the MICs for these different antibiotics. So interesting. Just another system you can use to assess effectiveness of antibiotics. Now, this would be, again, to a specific bacterium. It gets more complicated in you and me and in your kids and grandpa and grandpa, because we're not dealing with a single bacterial species, right? We're dealing, as we learned in one of the chapters, with hundreds and hundreds and hundreds of different species of bacteria. Right? And then this introduces another concept called the therapeutic index, which looks at the toxicity effect as well as the minimum inhibitory concentration. And they basically assign a therapeutic index value to the drug, as it mentions here. The higher the value, the better, the more safe, desirable the drug is, called the therapeutic index. And I think that wraps up the end of the chapter 12. We have about 6 or 7 minutes left. Any other questions on 12 or what we've been talking about? I just happened to flip to page 390, 391. There is a really cool Making Connections box entitled, "Are We Losing the Drug Resistance Race?" And it talks about drugs and animal feeds, worldwide drug resistance, and bad bugs, no drugs. I hope you're reading these Making Connections because they really are, I think, really interesting and pertinent to what the chapter is talking about. I think the case studies also, or the case study that is introduced at the beginning of each chapter and usually wrapped up toward the end of the chapter, are just nice little snippets. They're just really great introductions to, you know, what that chapter is going to be talking about. I hope you're finding those interesting. I mean, you guys in nursing, you do a lot of case study stuff, don't you? Or some case study stuff?
Yeah, we do those.
Yeah. So just to review real quick, the quiz then on Thursday over sections 12.4, 12.5, and 12.6, which is kind of what we wrapped up talking about, right? The last 25 minutes or so. You should be trying to get into 13. I will have the remaining third Zoom lecture for 13 done. My goal is tomorrow afternoon before I leave work. If it doesn't get done then, which I'm going to try to get it done, it'll definitely be done by the end of the day Thursday, because I want to stick with the schedule. And then again, next week, we've got the third exam over 10, 11, and 12. And then we're really looking at wrapping things up, because really after this week, we only have, I think, 2.5. Yeah, only 2.5 more weeks of the semester, and then we're done. And then you'll be so sad to be done with micro. Any last minute questions? Okay, I'll be here for another 5 minutes before my afternoon lab starts. So have a great rest of your day. And I'll see some of you in a few minutes. Otherwise, I'll see the rest of you guys next week.